Welcome back to day 2 of Innovations in Disease Modelling 2023. I'm Caroline Clark, conference producer at Technology Networks, and I'm here to introduce our first talk of the day. Bioengineering a vascularized skin on chip for viral immune research and antiviral drug evaluation. I'm pleased to welcome Jia Zhu as your presenter. Dr. Zhu is a research associate professor in the Department of Laboratory Medicine and Pathology, University of Washington School of Medicine, and associate professor in the Vaccine and Infectious Disease Division at the Fred Hutch Cancer Center. Her team has recently bioengineered a 3D vascularized biometric skin-on-chip device that is capable of immune cell and drug perfusion as an alternative platform for deciphering complex and dynamic post-pathophysiology to HSV infection and for preclinical evaluation of antiviral drug efficacy in humans. A warm welcome to you, Jia. Following the presentation, we will have a Q&A session and we will welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. You can also use this feature to connect with attendees and discuss the talk, so feel free to take advantage of the chat system by introducing yourself and sharing your thoughts with Jia's presentation. If you experience any technical difficulties during the presentation, please request help using the technical help icon in the menu on the left-hand side of the player. Now, without further ado, I will hand over to Jia to begin her presentation.
All right, thank you, Caroline, for this nice introduction. Today, I'm going to talk about bioengineering a vascularized skin on chip for viral immune research and antiviral drug evaluation. You probably already know herpes simplex virus. It is one of the most common pathogens in humans, affecting 2/3 of the world population. It affects both developing and developed countries. The virus is a large double-strand DNA virus with a genome size of 150 kilobase pair and encode more than 80 viral proteins. Although there are two subtypes, HSV1 and HSV2. Although they share about more than 80% genome sequence, they differ in serology, preferred route of transmission, and clinical manifestation. HSV1 typically limit to cold sore and keratitis, while HSV2 is the main cause of genital herpes, a sexually transmitted disease. HSV is acquired through close contact with an infected person. At the initial infection, HSV enters the body through breaks in the skin and the mucosal surface and start lytic replication in the epithelial cells at the site of entry. Viral particles are then taken up by the free sensory nerve endings innervating the tissue and transport through retrograde axonal flow to the nucleus of the sensory neurons, where it establishes latent infection for the lifetime of the host. Under various emotional and physical conditions, such as stress, fatigue, and menstruation, sunburn, or neuronal injury, HSV can reactivate from latent state. And the reactivated virus can travel through anterograde axonal transportation back to the skin and mucosal surface, where it close to the original infected site. Virus shedding can occur in the form of ulcerative lesion or completely asymptomatic with no symptom at all. So the mucosal virus shedding leads to transmission to sexual partner, and in some cases, it can be transmitted vertically from mother to infant at birth. So we focus on study genital HSV2 infection because it's it's a disease has a very high impact on public health. HSV-2 infects 500 million people worldwide. In US alone, 1 in every 6 women aged between 14 to 49 are HSV-2 seropositive. The virus causes neonatal herpes that often result in severe and lasting neurological impairment. HSV-2 is a known risk factor for acquiring and transmitting HIV. And importantly, there's no vaccine available. All the candidate vaccine that showed the great promise in the mouse model, they all failed to protect in human clinical trials. So this highlights the importance of understanding protective immune response in the human host. And clinically, HSV herpes infection has very wide spectrum of disease outcome, ranging from complete disease-free to sporadic short-lasting recurrence, and to very severe and frequent ulceration that requires daily suppressive therapy. So this large variation in disease expression allows one to dissect the success or failure of an immune response in humans. And people say that herpes is the true love and forever, once has, never forsakes, because the virus has ability to establish latent infection in the nerve system for the lifetime of the host. So it plays hide and seek with the host. So this lifelong infection provides a natural platform to study the evolution of the host immunity and also the formation of tissue-resident memory. But the good things, and it's from a practical point of view, genital skin and mucosa is accessible for frequent examination, and it's tolerable for biopsy studies. So this provides an invaluable opportunity to study the battlefield where things really matter. Therefore, we spend our careers really trying to understand the natural infection of genital herpes, especially its immune response in the human skin. So the research focus of my lab is to deciphering this protective immune response at the site of HSV-2 reactivation in human skin. As you can see in this tissue section of an ulcerative lesion, viral replication occurs at the skin epidermis in the presence of massive immune infiltrates, as indicated by the CD45 positive immune cells showing green. So we ask ourselves, what are the immune subsets that are present? How do immune cells spatially relate to the infected cells and their temporal dynamic change over time? What are their functions in immune protection and regulation? How we identify their unique gene signature? And how do T cell immune landscape look like? How diverse are the T cell repertoire in the tissue, and what is their clonal distribution? Can one identify tissue-resident memory T cells through T cell repertoire sequencing? And how do immune cell crosstalk and communicate with each other? Can we identify correlates of protection that are associated with asymptomatic HSV reactivation? And how the tissue microenvironment shape local immune response, especially the roles of keratinocytes and peripheral nerve play in HSV pathogenesis. To study those questions, we use sequential biopsies. So we collect 3 millimeter punch biopsies, either during a natural course of genital herpes recurrence at the time of active lesion, newly healed, and then 2 weeks, 4 weeks, and 8 weeks post-healing, or during a secondary antiviral therapy. In this case, we collect sequential biopsies up to 20 weeks post-healing. We also collect normal skins from arm or at contralateral site as a control biopsy. So because our material, the punch biopsy, is only 3 millimeter in size, so we have to apply and adopt a variety of tissue-based technology to maximize our understanding of local immunity in skin. This includes to use multicolor confocal microscopy, transcriptional profiling, high throughput TCR receptor sequencing. And in combination with all this, we develop cell type specific laser capture microdissection to isolate individual cells of interest while maintaining its original spatial distribution and native physiological state. So with those technology tools in hand, we actually show that CD8 T cells, including HSV-specific CD8 cells, persist in tissue long after lesion completely healed, and they are contiguous to the sensory nerve endings, which is the gateway of reactivating HSV. In fact, also even during a secondary suppressive therapy, both CD4 and CD8 T cells persist in tissue and interact with dendritic cells that form inflammatory manures in tissue. And this is the potential cause of increased HIV transmission and acquisition, as they provide immune targets for HIV. And we also have shown that CD8 alpha T cells are the dominant tissue resident memory T cells in skin after herpes reactivation. And they perform immune surveillance and the rapid containment. They are the force behind asymptomatic reactivation. And we find that high CD8 effect to target ratio and the focal enrichment of CD8 alpha T cells, and also the rapid recruitment of CD8 alpha beta cells, and also increased cytotoxicity are associated with host effective control before virus spread and form lesions. We recently showed that HSV reactivation promotes peripheral nerve growth through IL-17C and IL-17RE pathway between keratinocytes and the peripheral nerve fibers. While this work has advanced our understanding of HSV pathogenesis in human, it also has its own limitation, like any other model systems. So here, I just want to show an example of asymptomatic HSV reactivation and how the immune response looks like in tissue of asymptomatic reactivation. As you can see, indicated by the arrows, we saw limited viral replication, viral gene expression in keratinocytes in tissues of asymptomatic reactivation. And this tissue was taken 8 weeks post-healing. And we, using sensitive PCR detection, we can detect there's about 75 copies per million cells. And in this tissue, you can see there's multiple CD8 T cells form close interaction with a single virally infected cells, showing red. And we saw this increased number of cells and the increased amount of preference are expressed in those CD8 T cells. So the fact that CD8 T cells express perforin granules further indicate their cytotoxic effect function and really suggest their role in immune surveillance and rapid containment. So the biopsy work has really advanced our understanding in HSV pathogenesis in humans, but it also has its own limitation, as I mentioned. So assessing to human tissue is limited, both in size and numbers. And the biopsy we collect is only about 2 to 3 millimeters in size and only 1 per disease stage during an infection. So it is hard to perform genomic, proteomic studies that needs a lot of material. And it is also difficult to capture critical early events in disease progression. Even though the earliest time points we can get to capture the pustule lesion is more than 48 hours. So there is also a lack of accurate information on the kinetics of key immune cells. It's easy, and also it's easy to be criticized as descriptive, and we need to develop new models for mechanistic study. Also, the high prevalence of HSV1 and HSV2 in the global population, plus the suppressive antiviral therapy, is facing resistance, and there's no cure, no available vaccine for HSV. All these limitations and demands motivate us to bioengineer biomimetic 3-dimensional human skin on chip with a built-in microvasculature as an alternative platform for mechanistic study and therapeutic intervention. So our goal is to bioengineer 3-dimensional biomimetic human skin that recapitulate natural skin architecture and function. This requires a stratified epidermis on top and a dermis at bottom with connective tissue for structure and mechanical support, and a blood vessel network for circulation. So a desired engineered skin should retain the protective barrier function and is responsive to infection, and the vessels are permeable for drug delivery and treatment. So this allows us to monitor HSV infection and immune response in real time, as well as to assess drug efficacy and other therapeutic agents. So here are the photos showing how the device look like in real life and in fluorescent confocal image. So the H&E staining indicate the engineered skin has a typical histological features of a stratified epidermis, including a basal layer and a differentiated spinous layer, granular layer, and a quantified layer. Immune fluorescent staining also confirm key marker expressions of differentiated epithelial layers, such as keratin, and for differentiated keratinocytes forming spinels and the granule layer, and the CK14 for basal keratinocytes. We also show basement membrane formation underneath the basal keratinocytes and the fibroblasts in the dermis. This pattern of expression is similar to the natural genital skin we collect from patients. So the confocal image showed differentiated stratum bacille from all other stratum layers, and showing the outmost layer of the stratum corneal with no nuclei and no organelles. So we have examined the integrity and the permeability of the microvasculature. We perfused the system with 40 kilodalton fizzy labeled dextrin to check any leakage and calculate the permeability. They are pretty close to the small venule that collecting blood from capillaries. We also use confocal microscopy to examine the structure of microvascular network. The cross-section area of the microvessel show the oval-shaped blood vessel-like luminal vasculature. Also, the actin filament aligned along with the median flow indicate the vessels are responsive to the mechanical flow shear stress. We then infect the skin chip with HSV K26 strain, which is a GFP expressing virus. Inoculation of HSV direct on top of the skin resulted no infection, indicating the barrier function of the stratified epidermis. We had to make a mechanical punch to create physical disruption to facilitate infection, to allow virus access to susceptible cells. Infection is mainly occurred near the epithelial rupture. We also see viral spread occurs in basal keratinocytes, in CK14 and the positive and the CK10 negative keratinocytes. The HSV infection in the skin chip also induce morphological changes, such as the formation of multinucleation, enlargement of cell nuclei, and the chromatin margination, similar to the characteristic that we have observed in human HSV ulceration, showing on the right side. To evaluate the roles of keratinocytes differentiation in HSV infection, we infect epithelium with HSV at several differentiation time points. Undifferentiated replicating keratinocytes monolayer was the most susceptible to HSV2 infection. Even 1 day after culture in 3D differentiated media resulted in 70% reduction in GFP production. After 7 days differentiation, almost non-keratinocytes were observed infected. The results identified basal keratinocytes are the susceptible for HSV. Non-replicated differentiated cells are resistant to infection. The data also highlight the importance of intact barrier function in preventing viral infection. When we tested whether HSV infection in the 3D skin chip can respond to acyclovir, which is the common antiviral drug prescribed to patients with severe herpes, we perfuse acyclovir at 20 micron concentration at different times, either 24 hours before viral infection, or perfuse at the same time of infection, or 24 hours after viral infection, or with no acyclovir at all. The viral infection lasts for 48 hours. So as you can see, the perfusion of acyclovir inhibit HSV infection in a dose-dependent manner. We also perfuse acyclovir at a 20 micron concentration, 24 hours before immediate or 24 hours after viral infection, and investigate acyclovir activity at a 48-hour post-infection. The timing of treatment had a significant impact on acyclovir treatment efficacy. Perfusion of acyclovir 24 hours before infection or immediate after infection showed much more potent inhibition than perfusion 24 hours after infection, as reflected both on the number of virally infected cells and the intensity of GFP expressed, highlighting the importance of taking early treatment. There's no significant difference whether the skin chip was pre-treated for 24 hours or treated right at the infection. So in conclusion, this skin-on-chip device can recapitulate effective antiviral activities in vitro using clinically proven therapeutic dosage. So the 3D skin-on-chip platform showed the great potential for clinical efficacy testing and for PK/PD modeling. To validate whether the endothelialized blood vessel, like vascular network in the skin on chip, is competent in mediating immune infiltration in response to skin infection, we perfused fresh isolated neutrophils through the microvascular network after epidermal infection of HSV1 K26 infection. 1 hour after HSV inoculation, we perfuse neutrophils and monitor its transendothelial migration by live cell imaging. Neutrophils were shown to rapidly attach and adhere to the endothelialized vessel wall within minutes of perfusion in the skin chip with HSV infection. Neutrophils accumulate along the endothelial wall and then transmigrate efficiently through the endothelial wall in the dermal space. Even 4 hours post-perfusion, significant amount of neutrophils migrate out of the vessel in response to HSV infection. So at the 20 hours post-infection, the skin chip was fixed and stained and imaged using confocal microscopy. So quantitative analysis indicate the majority of neutrophils were extravasated into the dermal space and migrate upward, with a small portion of them reaching the infected epidermis, indicating a direct migratory response by neutrophils. Reconstituted image confirmed that HSV infection mainly occurred in the skin epidermis, where while neutrophils migrate upwards toward the site of infection and form close interactions with HSV-infected cells. And we also be able to detect pro-inflammatory cytokine expressions in the skin on chip system. We detect IL-8, IL-6, and RANTES expression, but there's no TNF expression can be detected in the system. So this indicates that HSV infection can markedly induce IL-6, IL-8, and the RANTE production. And notably, that IL-8 was the most abundant cytokines detected at the baseline and in response to HSV infection. The predominance of IL-8 before and after HSV infection provide a mechanism supporting rapid recruitment and massive infiltration of neutrophil to the site of HSV infection. And we compare to the HSV infection in humans. As you can see, neutrophils, as indicated by CD15, they migrate toward infected site, which is occurred at the skin epidermis. And there is abundant IL-8, IL-6, and RANTE expression detected in skin, while there's very limited TNF alpha can be detected in the skin. So this comparison really confirmed that elevated expression of IL-8, IL-6, and RANTES in HSV lesions compared to control uninfected skin. We then further confirm that neutrophil transendothelial migration is dependent on IL-8 expression. So in this case, we use an IL-8 neutralizing antibody perfused through the network, and then infect with herpes. And we can see with the antiviral anti-IL-8 perfusion, there's dramatic reduction in the neutrophil attachment to the vessels, and there's almost none migratory neutrophils can be detected in the outside the vessels in the dermal space. And on the right side, you can appreciate using RNA scope coupled with protein detection, we can show that IL-8 RNA transcripts was largely expressed by K10 positive differentiated keratinocytes in the skin lesions. So this really highlights the role of IL-8 in in directing neutrophil migration and extravasation. So in summary, we have bioengineered a three-dimensional skin-on-chip tissue device that consists of a epidermis, dermis, and the microvascular network. The bioengineered skin showed the characteristics of stratified epidermis, and the microvascular network forms functional lumens with endothelial cells, and we have shown that the endothelial integrity and the angiogenic capability, and this system is suitable for perfusion. And this bioengineered the skin show the similar characters as natural skin, and it's susceptible for viral infection. And also, this bioengineered skin tissue can respond to antiviral treatment, and is competent for immune cell transendothelial migration. So we conclude this skin-on-chip can serve as an alternative platform for dissecting complex viral host interactions in humans, and for accelerating therapeutic discoveries. I would like to thank the people in the lab contribute this work, particularly CJ Sun, who is the leading author of our Nature Communication paper, and all our collaborators and the funding resources. And I thank you for your attention.
Thank you very much, Jia. That was a great talk. And as with all the other talks, we are likely to going to have lots of questions from the audience. And I can see that we've received some already. So I'll move straight on to the Q&A now. So your first question is, does this skin on chip contain PDMS?
Our skin on chip do not contain PDMS. Because we use plexiglass for it. The only place we use PDMS is to make the stamp, which is to generate the geometry for our system. But in our skin on chip itself, we don't have any materials that is made from PDMS.
That's great. Thank you for that, Jia. We'll move on to your next question. Is the device suitable for drug preclinical testing?
I think we have the potential, especially our system is free of PDMS, which is widely used material in microfluidic system, but it is also has a well-known propensity to absorb small molecules and interfere with drug discovery. So we think our system might be a good for the preclinical testing, because it doesn't have the PDMS, and we have already demonstrated they have venue-like permeability, so the drug will be able to get into the tissue, and for modeling the drug efficacy, delivery, and also kinetics.
That's great, thank you. We have time for one more question, and that is, what is the application of this in personalized medicine?
Yeah, so I think currently we are working toward that direction, because right now the system can use commercial available cell sources to make this and study the innate immune response. But we are currently in the development of obtaining HLA matching cell components from the a small biopsy tissue. So from the same person, we can also get PBMCs. So this will allow us to make personalized skin on chip to study disease severity. As you can recall in my introduction, herpes does have very wide spectrum of disease. Some people is completely asymptomatic, while others have very severe disease. So we hope by using personalized skin chip, we can actually study the people who have completely without symptom, like asymptomatic HSV, as compared to people who have very severe HSV disease. So we are quite excited about that. Yeah.
That sounds great. That really does. Great work going on there. So, and I'll thank you very much, Joe. And unfortunately, that is all the time we have for today. Any questions that we didn't have time to get to will be sent to the speaker after the event. And as with all the other talks in the event, this talk will now be made available to view whenever you want for the next 30 days. So that just leaves me to say thanks again, Joe, and we'll be back soon with the next talk.
Thank you. Thank you all.